My guest today is Dr. Marcela Uliano-Silva, who's Senior Bioinformatician at the Wellcome Sanger Institute. Her research interests are mechanisms of genome evolution, the improvement of genome assembly pipelines, and actions towards inclusion and diversity in science. Welcome, Marcela.
Thank you. It's good to be
Yeah,
here.
thanks for doing this. So I want to start with your 2020 paper, towards complete and error-free genome assemblies of all vertebrate species. You say high quality and complete reference genome assemblies are fundamental for the application of genomics to biology, disease, and biodiversity conservation. However, such assemblies are available only for a few non-microbial species. So, and you say here to address this issue, the International Genome Consortium has worked over a 5-year period to evaluate and develop cost-effective methods for assembling highly accurate and nearly complete reference genomes. So before you get to the details of this, Marcela, could you talk a little bit about the International Genome Consortium, who is involved, and how that has been organized?
Yeah, sure. So yeah, I think that was the first thing I would say is that it is, I mean, I am in this paper, but it's a huge effort for a lot of different scientists in this consortium called the Vertebrate Genomes Project. So which is basically shared by Eric Jarvis from Rockefeller University in New York. So the history behind it is basically that he studies vocal learning, and there are a few birds that are able to learn and also to create new songs, because a lot of birds they copy and they have this specific song from generations on, and they always sing the same song. But a few birds are able to create new songs and pass it on to their offspring. So basically what Eric does is that he studies the evolution of language in humans, the genetics, and compares also with birds. So he studied this peculiarity in specific birds. And then from the first generation of sequencing genomes, which was basically sequencing short reads DNA, I can explain exactly or very basically why one technology causes problem in terms of the other. But just long story short for now, I can state that short reads cause more problems in assembly. So assembly is the reuniting of all of this DNA to form the chromosomes of the species, right? When you don't know how these chromosomes look like. So he was doing this organization of chromosomes, so this genome assembly for birds, using a technology that uses short DNA sequences. And this is prone to give us a lot of errors. Is that if we had a lot of misspellings in the sentences of a book, for example. So as using these genomes to study these birds with these essential errors, he got really frustrated and he was like, look, we have been trying to improve genomes for a long time. So let's do all of the vertebrates because we really need to have this in very high quality. This is because...
Just one quick question, Marcela. So the short genome sequences, so you're doing it in a short steps because of the technology constraint, or there's some other reason why that's the case?
Yeah, so basically, since the sequencing of the human genome, which was sequenced with a technology developed by the person that my institute is named after, which is Sanger sequencing, during the development of the human genome, which is using this Sanger sequencing, which is we can sequence long stretches of DNA, but in a much less scaled manner. So you would really use like 3,000 scientists to do the first version of the human genome. So during that period, lots of companies and academia were developing new methods to scale up and make the sequencing of DNA much faster. And one of the technologies was basically to sequence shorter read fragments. So you just basically take the DNA of a species from a cell, you chunk it up in pieces, because it's very difficult for the scientists in the lab to extract an intact DNA from a cell. So usually it breaks. So, and then you do a little bit of more breaking to get like a uniform distribution of DNA. And then you would put adapters on this like whole genome sequencing, as we say, and you would sequence with these machines that were sequencing shorter fragments. Soon we came to realize that because DNA genomes have a lot of repetitive sequence, And when we try to put these pieces together, in a method that we call de novo, which is just basically like a jigsaw puzzle. So we sequence different portions of DNA redundantly, and they have slight overlaps between them. So basically, if you have a redundant coverage of specific regions, you can put them on the top of the other, and then you form a larger consensus. The problem is when you have a repetitive sequence in the DNA, algorithms get very confused and they pile up all of the DNA one on the top of the other. So we basically soon came to realize that short sequences were not very, they were very difficult to resolve the repeats. So companies and academia again start developing more and more methods and machines to sequence as long sequences as we can. So then we have like a larger jigsaw puzzle piece that in the overlaps are less prone to error, right? They are more reliable. So this is basically the transition between all these bird genomes and other genomes that Eric and others and I and others have been using in the past, and we are finding systematic errors on these genomes. Even though these genomes help us to advance science a lot, there was a lot of things we can think as like a old photograph film. There was a lot of pictures there that were not showing because the whole film was not assembled, right? So we basically gathered together in this consortium and many others. So, and to redo and to make the final reference genomes with this new technology that is in place now.
You were talking about, is it the zebra finch, the bird with songs and so on? And so what's the connection there between the bird and the human?
Oh, that you would need to talk to Eric because this is his side of the specialty. But it was basically, so the fact that he was finding out of these errors in the genome kind of brought him to have the idea of gathering a consortium of scientists to do all of the vertebrates. And I was actually studying the sloth genome. Do you know the sloth? I never know how to pronounce that very well, but it's the hang mammal from South America. And I was studying this the species in Germany at the time, doing a high quality genome, and then I joined the consortium, and we have been working together ever since. Now I am in Cambridge, working at the Sanger Institute, where we have another consortium called the Darwin Tree of Life Project. So because basically now we are in a state that we can sequence very high quality genomes with almost all of the information present in this highly contiguous chromosomes. So we have these different consortiums. So one of these consortiums is the Vertebrate Genomes Project, which is taxa-based, right? It's to sequence all of the vertebrates. The Darwin Tree of Life project is regional based. So it's basically a consortium to sequence all life in Britain and Ireland. So it's 60,000 species. It's those like huge efforts to sequence all life on Earth, basically.
So there's the Earth BioGenome Project?
No, so the one that I am primarily employed by, let's say paid by, is the Darwin Tree of Life project.
Oh, okay.
And the Earth BioGenome Project is basically the umbrella that encompasses all of these other projects, right? So it's like the Vertebrate Genomes Project, the Darwin Tree of Life Project. We have another project, which is the African BioGenome Project. We have the ERGA, which is the European Genome Consortium Atlas. So all of these consortia are trying to basically not reinvent the wheel, right? They're gathering scientists together to work on these massive efforts to sequence or regional genomes or taxa-related genomes.
So this is a consortium of academic institutions and individual researchers who all are involved in the consortium.
Yeah, and this also comes to my diversity and inclusion work, because we want to have as many people as possible involved. Because if we do want to sequence all species on Earth for the benefits of science and for the benefit of everyone and conservation and everything, we want to have as many people as possible involved. And then, of course, some big institutes wrote the first grants and got the money, and they have infrastructure. So the Darwin Tree of Life, for example, at the Sanger Institute has a huge infrastructure of sequencing, right, and a budget to pay scientists to do it. But we are also accepting collaboration, so people can send us samples, and we do the sequencing, and it's open access. And the African BioGenome Project and the Earth BioGenome Project is also has a centralized, let's say, governance. But again, we have this committee in diversity and inclusion to talk to other people that want to reach us and want to their species to be part of our project, and we want to do this as inclusive as possible. So it is a group of institutes, research scientists, and we want to grow as much as possible. Some other things are, for example, some species are like indigenous, species of indigenous importance or things like this. And we don't want to just go and interfere and basically collect a species and sequence anyway without taking all of that background and all of that importance status for a specific community out of place. So we are also having scientists and sociologists and anthropologists to like be involved on this. So then we do this as inclusively as possible and not just like gathering samples and sequencing without giving back to the people there, to which this species can be precious or important.
Yeah, so I'm thinking, Marcela, so this is like building a library, and there are a lot of books in the library, and the books has a lot of pages, a lot
Oh
of
yeah.
text. And you're starting sort of from scratch because we don't really have completed a book in many ways, right? So the goal here appears to be to complete a large library. And what do you think? What is sort of the end objective? Suppose you completed this project and we have reference genome of every biological entity on Earth, let's say. I don't know how long that's going to take, but suppose we have it. Where will you go with it? But how does that information be used?
Well, I mean, it's also on the way there. There's so much happening already, right? So first, so we have like a massive system in place, employing a lot of people working right around the world, which is the first thing, I guess. And on sequencing, we learn so much. So, for example, I have developed now a method to do an automated assembly of mitochondrial genomes using this new technology called PacBio Hi-Fi that didn't exist in the past. So this is one goal, one point, which is developing already new software and new technology to deal with all of this data as we go along the way, because it's a massive amount of data, right? The other thing, which is like for me, which is like for my egoistically, let's say my with time, I mean, since the sequencing of the first non-model organisms in the human genome, and then we started introducing non-model organisms, we have learned so much about evolution. And there were genome, like some shapes of genomes that we thought would be like typical animal, and then as soon as you sequence some other taxa or some other groups, you understand that they are not typical at all. So one example I can give you is that there are papers, which is very interesting to read. In the 90s, when the Human Genome Project started, there were papers stating like, oh, we probably have 100,000 or 200,000 genes because we are so much different than other animals, and we're going to find this in the human genome. And then when we sequenced the human genome, we found that we actually had the same number of genes as any other as much of the other taxa in nature. And then we'll go like, wow, why is that? And then you start looking at the genome and you see that the way that the information is expressed is actually what makes us different. There are a few specific gene families, and the way that that information is expressed is what brings biology together. But then one part that was really missing that we are being able to recover now is centromere and telomeres, which is this repetitive heterochromatic parts of a genome that until now were not uncovered. So we also want to understand how these parts evolve, and etc. And even considering now that we know a lot about the human genome, for example, because we have been understanding so much, there is still a lot that we don't understand. There is a lot of genes, which I can say it's like this reviewed photographs on this film. So there are some photographs we can see and some we cannot see, let's put it this way, So the ones that we can see, we also don't understand the image very well yet. And this is basic. So if we consider, as we do, that we are all connected and we all have a common descent, we are all linked, basically what happens is that throughout evolution, so DNA is the genetic material of all life on Earth. So basically, there are degrees of differences along the tree of life, but we can basically also find a lot of regions of conservation between among these genomes. So one of the other like important things for evolution is that we want to basically align all of the conserved regions among different taxa, and then we can see what is conserved and what it's not, and can relate it to phenotype, for example. So things that, you know, all of them have the digestive system that is this way, or so on, like specific traits, and we can see the importance of specific conserved regions for humans or for the biology of other organisms. In a long story short, there are still blank pages in how the human genome works and how health works, because we don't have this other in-between genomic evidence and analysis that it would really help us understand human health better. And then when we talk about human health, which would be the top of the chain, let's say, the in-between will help us a lot to also understand peculiarities, the genetics that lead to peculiarities in specific groups, which would be able to help us to conserve them better if we would like, if we would need this information. So it's really not an end goal that we are going to have all of this library and then we are going to look at results. The results are coming in as the project is being developed, you know, which is really exciting. And yeah,
it's fascinating. So let me see if I understand this. So you mentioned that it's almost like the hardware of all the biological entities on Earth are similar, and it's really the software that makes a real difference. Is that a good way to think about it? Would you?
That's very interesting. Let me think. Almost. So yeah, like, because I mean, this is almost like epigenetics, if we'd say the software, because the hardware, we also have some power differences or some storage differences, let's put it this way. Okay, they're not necessarily all the same, but they would be all hardware. Yeah, so, you know, oh, that's a hardware, I can see it, right, which is DNA. But then they would have peculiarities, and then their connection with the software would be it would be a bit different. So maybe different languages, different ways of connecting both, and also a little bit of difference in the hardware itself. It's not only the software, but a lot of the software is involved in what creates species and how they exist on Earth.
And you mentioned epigenetics. I don't know if I can take this analogy in this direction, but so there is an operating system and there's applications on top of that, right? So I think of this as the hardware, as you say, the CPU, the memory, the storage, all are different in different entities. But they also have some level of operating system commonality, right? In other words, if the DNA is there, how the DNA expresses information, how it communicates, all those things are somewhat similar, right? And so I'm thinking, once you put this all together, Marcela, right from the beginning to the end here, are there any sort of, I'm thinking, wouldn't this allow you to design new biological entities in some ways?
Oh, well, people are already trying this. So, so interesting. It's possible, yeah, I would say. It will be possible. And the same happens with people trying to de-extinct species, right? So they are basically sequencing ancient DNA and basically trying to recreate them with the cells from related species that are alive today, like extant species. So yeah, it's possible. There is a lot of ethics to be discussed in there.
If you can bring the dinosaurs back, that would be interesting. But it might also, would it show some sort of missing links in the sequence? In other words, we see X and we see Z, but we don't see Y. But you could logically conclude Y would have existed because X to Z transition would have gone through Y. So do you think we would find some missing links that we haven't found yet?
Oh, that's very interesting point. Well, one would think it's not directional evolution, so it's not necessarily that if you have X and Z, you would need to have Y in between. So this is something that we do a lot to try to see where evolution is going to take us. For example, like in cases of viruses, we would like to be able to predict mutations, for example, and like where mutations are more prone or not. So I think we can infer a bit, but it's not really my area. But that's very interesting question. It's possible we will be able to.
Yeah, mutations are quite interesting, right? I mean, it's very topical now with with COVID as well. And so would we get to a point that given the status quo, we can assign some probability that the evolution will, sorry, the mutation will take us into more potent organism or something along those lines? In other words, can we predict diseases in that way?
Yeah, well, so every organism operates in the laws of natural selection, right? So it's like the survival of the fittest. And then in epidemiology, I'm not an epidemiologist, but I can talk a bit about this. In epidemiology, they would say if you are very potent to kill your host, then it's not very likely it would be passed on, because that host will not be able to reproduce, and it would die, right? And then you would die with it. So for epidemiology, what epidemiologists usually say is that the disease, so the perfect disease would be the one that is like very mild, at least until it's passed along to another host. So, and you wouldn't kill the host very quickly, right? So that's why I guess we see flus and maybe the COVID, don't quote me on that, because we still need to, it remains to be seen, but it seems like it goes a bit more harmless in the to that human population, and then it will be kind of exist forever. So to be like more potent is actually against the laws of the survival of the fittest of natural selection. So we wouldn't say that, and not only if it was intentionally created.
Or we could, or let me ask you this, is it possible that we can say something is intentionally created with this type of data?
That's called a very question. Yeah, I mean, yeah, if you don't have any phylogenetic links with anything else, there are definitely possible genetic markers that could lead you to that hypothesis. Yes, to that possibility.
So I want to talk a little bit, I don't know anything about this, Marcela, but I want to talk a little bit about the technology. So you talk about here iterative assembly pipeline, scaffolds generated with all three scaffolding technologies. So could you talk, so this is software, right? So you're using machines to essentially go through data and put that together in some way?
Yeah, so it is basically, yeah. So briefly, you have like a specimen, lots of cells. Someone in the lab extracts the cells and extracts the DNA inside of the cells. So if we consider a human, we have 46 stretches of DNA inside a nucleus of a cell, which is the 46 chromosomes. So the DNA is not like a one linear big thing. It's 46 pieces, right? It comes from the beginning to the end for each of the chromosomes. So you extract that, and then so the lab person is extracting that, and then it tries to these days it tries to extract as integer as possible, as like without any harms, without any breaks on it as possible, but it's really like a mechanical thing to do that. And then they put a lot of adapters and things in like in a mix in all of the units that the cell also uses to sequence DNA, which is the DNA polymerase and all of the ACTGs to feed that polymerase to copy that the DNA, right? And this is basically captured by a machine, usually captured with light or a membrane polarization discontinuation, I guess, depending on which technology you're using. And then it's captured by a machine. So it's very interesting because for the long reads, you put it one single DNA molecule within a little pore, and you capture as each DNA is attached to the complementary sequence, and then you emit a specific certain light, and then the machine captures it. The interesting thing there is that the DNA polymerase is much faster. Our life is much faster in sequencing that strain of DNA than the machine is able to capture it. So then we have what we call insertions and deletions errors in the DNA sequence. So as bioinformaticians, then later on need to deal with that and probabilities of that sequence being correct or wrong in the computer, right? So this is just one type of technology that is in that paper. Then we have some other types of technology that we created to create what is called interactive DNA sequencing, because DNA, the chromosomes have different properties, as I said, in the different portions of it. So some are heterochromatin, like lots of repeats. Some are rich in genes, so the genes are a little bit easier to understand. And different technologies can help us get better the information of these different parts of the chromosome, let's say. So there is another technology that basically, instead of just trying to extract this very high quality long DNA, it takes the information of the 3D structure of the chromatin, because in the cell the chromatin is just like all not organized, and it touches itself. So we kind of freeze that information in time and then do a lot of lab work and transform it into information, which is called high-C information. And then it's just basically also helps us get this long DNA reads together in a better fashion, let's say, in related to the size of the chromosome, because it can gives us long distance information between long read sequencing. So it's to do these genomes, it's a game of having more than one type of technology sequenced. So having a very high quality sample, okay, first of all, so having a very high quality sample. And here we can already see a very big challenge for sequencing all life on Earth, because some organisms are very small, so it's very difficult to get good DNA from them. Some species are endangered, so you don't want to kill them or harm them, so you get DNA. So this is already a very big problem. So you should have very high quality DNA. You have to have these different types of technologies, and then you go to the computer, and then the bioinformaticians program different software and use different software programmed by others to calculate the probability of errors in the sequences. And then, as I told you, we sequence and cut in different with slightly overlaps between them. So they're not sequenced like from 1 to 5 and then 5 to 6. They're like 1 to 5, and then the next sequencing is like from 3 to the 10. So they have an overlap between 3 and 5, right? So we have this overlap. So basically we have like a big redundancy of a lot of DNA sequence, and then we do this overlap graphs, as we call, or the bridging graphs, which we would even like break these pieces of DNA in smaller pieces, and then we just basically try to see which like pieces match together and calculate the probability that they match together better here and not anywhere else. And then we just basically the computer basically outputs a consensus from there. And then it's interactive, because then it takes the other type of information that we sequence on the top of this one, and it corrects this one or just makes it better if it thinks it's correct. So it's just basically like lots of methods of sequencing and validation in each step to try to get as close as possible to the truth. Sorry, it was confusing. But it's just
No. So you mentioned repeats. on the DNA. So in short reads, you say that makes it more error prone because it's basically repeat. So um, why do you think that is? I mean, it seems such an inefficient design. Does it really have to repeat? Was it an error, the nature when it put together? I'm just
So
wondering why there's so many repeats on the DNA.
so this is interesting, isn't it? Because this is so people talk about the perfect design, right? Like the I is so perfect, and that's why maybe someone like creator created. But to be honest, evolution is just, it just happens. So the genome is actually basically a fight between the genes and some very old viruses. They are inserted in this genome. So these are the heterotransposons. So this is why, so this is one type of repeat, which we call red heterotransposon. So, and some are still active in some species genomes, which is very interesting. And they have this ability to self-copy themselves and try to go and insert themselves in another part of the genome. So this is very, very interesting. And there is also a hypothesis that epigenetics, which is these little things that go on the top of DNA, sometimes they just kind of like make the DNA not move. They were evolved to actually stop these heterotransposons from jumping everywhere around. So it's really interesting, you know. So this, and then evolution is just like in the middle of this thing, and then the thing that is most like, it doesn't spend a lot of energy, we can copy this and this still works, so we just keep copying it, so they exist there. We also find that heterotransposons are the source of innovation, which is really nice because sometimes they put themselves in the middle of a gene, they start expressing something else, and it can even create a new trait that eventually would create a new species. And some repetition is just some whole genome duplication events, again, because life is just random. So there was a time, there was a whole genome duplication for vertebrates, and then it wasn't very costly to keep copying this, and that exists. And actually, when you have that extra copy, then you can again put pressure on it, like different environmental variables would put pressure on that DNA, and it could even create something else. So it's really like a balance of randomness with pressure that creates new species, and just it's just like a mess all around. It's not a
So,
perfect design.
yeah, so that's what I was wondering, Marcela. So from a data perspective, one could argue that it's completely random, but I also wondered if there are preferences. Um, so if you take data, the initial conditions, do you think there are some preferences of how it evolves? It's not completely stochastic. Is it sort of guided stochastic?
Yeah, yeah.
Yeah.
Yeah, it's just not completely deterministic guided, but it for sure biochemical and specific bonds and chemical bonds would drive possibilities and probabilities more to one side than the other. Yes.
And so would that mean that some configurations are not possible? If you have, preferential heuristics driving evolution, some combinations may not exist, right?
Well, I mean, so we don't know how many species there were. We only know, I mean, we know some existed, right? But we only know the ones that prevailed. So it's possible they wouldn't be optimal, let's put it this way. And then they wouldn't survive, or, if they would create something. And we also have mutations that cause harm, right? So because also the DNA needs to be copied first every day to produce new cells, because we lose cells and then we need to rejuvenate our cells, so DNA is always copied again. And then you know this, if we copy one thing many times, we can cause errors. And that's also what causes us to, like, as we age, it causes mutations that causes to get disease and etc. And we also have errors when we copy to produce gametic cells, right? Cells that are going to go and produce offspring and everything. So and yeah, so if we do interfere with a specific gene, which function is very tight and needs to be coded a certain way, then it will be harmful, and then we will not like produce offspring, or we won't be able to, won't be viable. So that for sure exists. Yeah.
I had another guest on, Marcela, a few episodes ago, And he argues that it's not random mutation and natural selection. He argues it's artificial selection. It is basically the DNA seeking to go to a more optimum condition given
Interesting.
the environment. And so, I mean, it's obviously up for debate, but that's a different way of thinking about it, I think.
But was he talking about a specific case or?
No, in general. And so, my understanding is the commonly accepted idea is that you have all sorts of random mutations happening, and the beneficial one will get selected, and those are the ones that we see over time. He argues that it is the organism itself artificially changing to counteract the environmental conditions. So it's not really random, he argues.
That's very strange because I don't think that the DNA seeks anything. So I would need to, have a read on what he's mentioning. So in the case of humans, for sure, we have culture, right? So culture makes us act against what would be natural, let's say, in nature in general, right? So we would, so I guess, so there is like a fossilized bone of a human femur that was healed, now I don't know, but one of the first human communities that existed. And then they basically found a sign of humanity in there, because then someone would need to have fed that person, carry that person, and help that person to survive, because like other species would just probably left that person behind and then keep going and keep hunting and gathering because they cannot afford to have that person together with them, right? So what I'm trying to say is that humans basically do things against their own will in a way. We put ourselves in fire for like world peace and things like this, right? So this is also why when people say that they committed like harassment against women based on their instincts, it's pure bullshit, because we are much more culturate animals than we are instinct animals. So if he was referring to that, to humans, maybe he has some point, because the way that we live now, it changes our life so much that we are changing the path of natural selection. We are not just going to the specific, because we have antibiotics, we live much older, we have kids older. So we introduce all of this series of differences that can, have an impact on how our species are going to evolve. But for other life, I would say natural selection is the main force of evolution. Yeah.
Yeah, so I want to finish up with, I know that you do a lot of work in this area. So you have a blog entry, Scientific Excellence in Sequencing All Life on Earth Depends on Justice, Equity, Diversity, and Inclusion. So you want to talk a bit about that? So, uh, so you say life program at the Wellcome Sanger Institute, Genomes, Vertebrate Genomes Project. We talked about that global invertebrate genomes, genomics alliance, the Earth BioGenome Project. You talked about all of that. United over 5,000 unique attendees. So this is a conference that happened?
Yeah, so there's a conference called Biodiversity Genomics. We will have that again in October this year. It's free and it's online, and we intend to have it online forever, because I guess, I mean, it's not the most exciting, but I guess it's the most inclusive, because we can have students and people from all around the world. And this year we are going to have it in 5 different time zones. So, and then people from all the world can join, and then we will have the sessions recorded, so they can post questions even in advance if they want to watch a session from another time zone. So I can send you details when we have it, but we are going to open it for registration like in 1 or 2 weeks, very soon. Yeah.
If you send me the link, Marcela, I can put that on or when I publish this, I can put that link in there so people can.
Okay, that would be great. Okay, I will do that.
Excellent. Yeah, this has been great. Go ahead. Sorry.
Yeah. No, I think, yeah. So just to say about the diversity and inclusion. So we have this committee on the Earth BioGenome Project called JEDI. Everyone likes that acronym, which is Justice, Equity, Diversity, and Inclusion. Especially for biodiversity genomics, which is our field in science. And, of course, it would include everything that we are very used to discuss about diversity and inclusion, which is gender diversity, different human group diversity, and etc. But we also want to, as we want to sequence all life on Earth, there is a key issue, which is basically try to avoid neocolonialism of sequencing life from tropical countries without acknowledging the sciences there, or just basically collecting all of the biodiversity or the sequencing species information, bring into rich countries and sequencing here and not explaining anything to anyone, right? So when we do things inclusively, it takes much longer and it's much harder. We have discussions, we have conversations because people have different points of view, but that's exactly where we want to be and we want to go. So we don't want to leave anyone behind. So this idea is to basically sequence all life on Earth with people involved and like respecting regional, cultural differences and how they want to deal with their data and involving the scientists on this. So also trying to promote as much as possible training and exchange between scientists in the global south and in the global north. So it is a very, very big challenge. So as Earth BioGenome JEDI committee, because we try to address all of these challenges, we have 16 committee members from all around the world. So we discuss weekly, monthly. We are publishing a first paper now on the issues on JEDI in biodiversity genomics. And we are open to hear from, so anyone that is really interested in having all life on Earth sequence and wants to participate and thinks that there is a JEDI issue to be discussed or is uncomfortable with some way that the projects are doing things, they can always come and talk to us because we are here to ensure that no one is left behind.
And the data ultimately will be public domain, right? The data
totally public. Yeah.
And so anybody could use it. There could be a lot of uses for it, I would imagine, but that also brings some privacy concerns potentially, right?
Yeah, so this is the thing. So especially for specific species from specific communities, if they don't want to, how they would, so yeah, they want to open their data. So then it needs to be discussed with specific groups on how they want to deal with that data. If they want to have the data open on request or all of this. So this is exactly what we want to ensure. Our policy is that we would like to have it as open as possible because we believe that science should be open, but at the same time without disrespecting specific ancient knowledge and privacy concerns that people have. So we would always have groups to discuss this without publishing anything or sequencing anything.
Excellent. Yeah. So this is in October. So I'll put a link. If you send me the link, I'll put that on the program. And yeah, good luck with it. It seems like a really interesting project.
Yeah, it is. It is. Thank you so much. It's a lot of work, but yeah, it's challenging.
Excellent. Is there? I guess the umbrella you mentioned, that's the Earth BioGenome Project, right? The EBP. Yes. And under EBP, you have different wings of genome sequencing going on, right, for different classes of biological entities. If you were to expand it, is there scope for expansion to? Are you missing anything in the current objective?
Yeah, I mean, we heard now that India has its own biogenome project, which is really nice. We try to get Asia, which is very large, involved as much as we can. So we do, and we also miss lots of Latin America. So we have Colombia. I am Brazilian, right? So I have a lot of collaborators in Brazil, but then the Amazon is also a part that is like underfunded for science. And then we know there are very good groups there and professors, but they lack funding. So we try to collaborate with them as much as possible. So there is a lot still to be built. And if people are out there thinking about starting consortium or talking to us to see how they would do this, there is a lot still missing for sure.
I would imagine life sciences companies are also quite interested in this, right? I don't know if GlaxoSmithKline is part of Wellcome to anymore, but won't they have a interest in this type of data?
Yeah, for sure.
And then the question is the economics. If the data actually becomes useful economically, then who holds the right, and all of those questions need to be answered at some point.
Yeah, but then it's so it would be a technology built up on the top of the genome, right? So the genome is public, and the knowledge. I mean, we will try to do the science with the genomes, but yeah, what comes after it, I guess, will be as long as it's so. Since the human genome, also we discussed that we don't patent any of genetic information for anything, right? So it's only things that come after it, I would say. Yeah.
Yeah. What is the just one last question. So what's the status of the genome patenting? I know that there were some conflicts that people rushed to patent some aspects of human genome, and then that was challenged. Do you know where it stands now from a patenting perspective?
Yeah, no, there is no patenting of human genome information. Just you need to build up a technology on the top of it. Let's say you use a gene to insert in a specific like liver cell, then you need to have it all changed, and it's a specific like a probe which does all of these things that goes into the liver and then goes in. But the sequence itself is free, so anyone would be able to look at it. So it's completely free.
Right. Excellent. Yeah. Great project. Thanks for spending time with me, Marcela.
Of course, thank you for having me. It was lovely to be
Absolutely.
here.
Thank you.
Okay.